Good morning, everybody. Come fast. So, are you all ready now? So, let's start our lesson. So, day 6, our topic is anatomy of flowering plants. So, anatomy is the study of internal structure. That is the study of internal structure of plants and other organisms. So, started with meristematic tissues. So, is it visible? Am I audible to you all? So, give your response. Started with meristematic tissue, the apical meristem. Meristem that occur at the always the tips of roots and shoots. And apical meristem, they produce the primary tissues. Next, axillary bud, So axillary bud present in the axils of leaves, and they are capable of forming a branch or a flower. Next, intercalary. Intercalary meristem. So meristem occur between the mature tissues. That's the intercalary meristem. That is, in grasses, regenerate parts are removed by the grazing herbivores. So mostly present in the grasses, that you know. If this tip part is taken by the herbivore, then it again divides and forms the previous part by the activity of the intercalary meristem. So apical and intercalary meristem are primary meristem. So remember this one. Apical and intercalary, they are primary meristem. Next, secondary or lateral meristem. Secondary or lateral meristem. They occur in mature region. They are present in mature region of roots and shoots. That means the fascicular vascular cambium, fascicular vascular cambium, interfascicular cambium, and cork cambium. Remember this one. So these are present in the mature region, and the example are, that is fascicular vascular cambium, interfascicular vascular cambium, and cork cambium. So these are the lateral meristem, present in the mature region. Next one, the permanent cell or mature cell. Permanent or mature cell. The cells produced by primary and secondary meristem become structurally and functionally specialized. This permanent cell, they lose ability to divide. Then the permanent tissue. So there are two types. That is the simple tissue, permanent tissues having all cells similar in structure and function. When the permanent tissues having all the cells similar in structure and function. So their structure and functions are same in simple. That is parenchyma, the collenchyma, and sclerenchyma. So if we discuss the three permanent simple tissue comparatively, so we can say parenchyma are major component within organs. They are major in the organs. Collenchyma, they occur in the layers below the epidermis. Very important. Remember this one. Below the epidermis, mostly in dicots. Sclerenchyma, it consists of long, narrow cells with lignified cell walls. That's the criteria. Lignified cell wall having a few or numerous pits. They have pits. Next, Cells are generally isodiametric and the parenchyma, they consist of cell with much thickened corners. Much thickened corners. And sclerenchyma, they are usually dead and they are without protoplasm. Are you still in sleepy mode? So everybody, so the walls are okay. Walls are thin and this is made up of cellulose. In parenchyma, due to deposition of cellulose, hemicellulose, cellulose, hemicellulose, and pectin. Sclerenchyma, they are of two types on the basis of their structure and development variation. That is the sclerenchyma fibers and sclerites. In the parenchyma, the cells are closely packed or have small, very small, if present, very small intercellular spaces. Very small intercellular spaces. In parenchyma, the intercellular space that is absent in parenchyma. And in sclerenchyma, the fibers, these are very much thick walled. Elongated and pointed ends and occurring in groups. And the sclerites, they are spherical, oval, cylindrical, highly thickened. These are dead cells with very narrow cavity. They have very narrow cavity or lumen. So where the sclerites are mostly present, found in the fruit walls, of nuts, the leaves of tea, leaves of tea, pulp of fruits, food pulp, and seed coat of legumes. What the function of the parenchyma? Can you remember? The function of the parenchyma, that is photosynthesis, storage, and secretion. In collenchyma, they provide mechanical support. Beside, some storage is also done by collenchyma. And the sclerenchyma is the main mechanical tissue that provided support to the plant. So let's move to the complex tissue. The permanent tissue, first one simple, and second one the complex. Let us complex tissues. So complex tissue, that means first the xylem. So xylem, we know this is the conducting tissue. For water and minerals from root to the stem and the leaves. These are mainly of tracheids, the vessels, xylem fiber, xylem parenchyma. Tracheids, a kind of xylem element having elongated or tube-like dead cells, and it has thick lignified wall and tapering end in the flowering plants. The main water transporting elements, that is tracheids and vessels. The vessel is a kind of xylem that is long cylindrical tube-like structure made up of many cells, each with lignified wall and large central cavity, and protoplasm is absent. So they are all dead part. Only the living part in xylem, that is the xylem parenchyma. In gymnosperm, so vessel is absent in gymnosperms. Remember this one. Xylem fibers are highly thickened walls. They are separate. They are septate and aseptate both. Xylem parenchyma, this is the only living component in xylem. Thin-walled cellulosic cell wall and store the food materials and other substances like tannins. Here an important information regarding xylem. Give me the answer. The first formed primary xylem. What is the name? First formed primary xylem? That is protoxylem, and later form, Ankita, later formed primary xylem. What is later form primary xylem? Tell me. Yes, metaxylem. So in stem, the protoxylem, lies toward the center. That means protoxylem lies toward the center, very nearer to the pith. The protoxylem in case of stem. And metaxylem lies toward the periphery. In case of stem, this type of primary xylem. So this arrangement in stem is called endark. Remember this one. It's very important to ask this type of question. So when the protoxylem are present nearer the pith, that means in the center, and metaxylem in the peripheral zone in stem, this arrangement is called endark. And in root, in case of that is present in stem, in case of root, the protoxylem are present in the peripheral zone and metaxylem present in the center. This arrangement is called exarch. So is it clear? It's a very important matter. Next part, that is phloem. In angiosperm, phloem is composed of, what are the components? That is sieve tube, sieve tube elements, companion cell, phloem parenchyma, and phloem fiber. In gymnosperms, they have albuminous cell. In gymnosperm, remember this one. Albuminous cells and sieve cell. In gymno. But gymnosperm lack sieve tubes, very important, sieve tubes and companion cell. So that lacks in gymnos. A mature sieve element has a peripheral cytoplasm and large vacuole. So, in case of mature sieve element, only peripheral cytoplasm with a large vacuole, that lacks nucleus. Next, now the same concept, first form primary phloem. The first formed primary phloem, that is protophlloem. Yes, and first formed or later formed primary phloem, that is metaploem. Remember this one. Now some important parts of tissue system. The tissue system. Tissue system. First one. The epidermal tissue system. It forms the outermost covering of the whole plant body, that is epidermal tissue system. Cuticle, that is absent in root. Epidermis, is usually single layered. Guard cells regulate the opening and closing of stomata. A few epidermal cells specialized in shape near the guard cell, they are called subsidiary cell. They are specialized in their shape and shape. The stomatal aperture, guard cell, and the surrounding subsidiary cell, these are collectively called stomatal apparatus. Remember this one. Stomatal apparatus. So what are the components in stomatal apparatus? The stomatal aperture, the guard cell, and the subsidiary cell. Stomatal aperture, guard cell, plus subsidiary cell, they are unitedly form the stomatal aperture. In epidermal tissue system, the root hairs, these are, remember this one, these are unicellular. Elongation of the epidermal cell, they help to absorb water and minerals from the soil. The trichomes, they are the epidermal hair. The epidermal hairs on the stem, they are usually trichomes or multicellular. They are branched or unbranched and soft or stiff. They have the properties of secretory. These trichomes help in preventing water loss due to transpiration. That is the another property. So after epidermal tissue system, the next, that is ground tissue system. So in ground tissue system, all tissues except epidermis and vascular bundles. So except epidermis and vascular bundle, all are ground tissue system. Parenchymatous cells are present in cortex, pericycle, pith, medullary rays in the primary stem and roots. In leaves, the ground tissue consists of thin walled chloroplast, and these are called the mesophyll cells. So remember this one. Now the vascular tissue system, that is xylem and phloem, they together form the vascular system. Here the important concept, when the open type vascular system, that means cambium present. This is the open type that present in Dicot stem. Frequently asked question, open vascular bundle, cambium present in Dicot stem. Closed vascular system, here the cambium absent. and they do not form the secondary tissues. Mostly these are found in monocot. That's an important difference between dicot and monocot. Radial type. Radial type of vascular bundle. Xylem and phloem with a vascular bundle are arranged in an alternative manner of different radius. In different radius they are present. In different radius present in roots. Radial type. Another type, that is conjoint. Conjoint type. Xylem and phloem are situated at the same radius. They are present in the same radius. That's the conjoint vascular bundle. Common in, these are present in stems as well as leaves. So open type, closed type, radial type and conjoint. Next, the dicot root. In case of dicot root, the endodermis comprises of single layer of barrel shaped. Remember this one. They have barrel shaped single layer endodermis barrel shaped cells without any intercellular spaces. The tangential as well as the radial walls of the endodermal cell have a deposition of water impermeable, waxy material, suberin, and that is called the Casparian strip. So that is called Casparian strips. Can you remember the Casparian strip? The tangential, remember this one, radial walls in the endodermal cell have a deposition of water impermeable, waxy material, suberin is there to form the Casparian strip. Here the suberin is there. Still, all tissues of the inner side of the endodermis is treated as the stele. Pith is small and inconspicuous. There is conjunctive tissue. Conjunctive tissue. The parenchymatous cells which lie between the xylem and phloem. So, in between xylem and phloem, the parenchymatous cell, these are parenchymatous cell, lie between xylem and phloem. These are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Xylem and phloem patches in dicot root. Remember this one, it's identical character. In monocot root, do not undergo any secondary growth, but in monocot, the special one, in monocot root, that is called polyarch. Note down this one, it's very important. Polyarch. This is called polyarch condition. Yes, Ankita, you are right. So dicot stem, the ring arrangement in case of stem of vascular bundles are there. It is the characteristic of dicot stem. In case of dicot stem, the vascular bundle are arranged in ring. They are always in ring form. Each vascular bundle in dicot stem, they are conjoint open and they have endark. They have endark protoxylem. In dicot stem, pith is the central portion of the stem, where a large number of rounded parenchymatous cells with large intercellular spaces are present. That is the important character of dicot. Monocot root do not undergo any secondary growth. The hypodermis consists of a few layers of collenchymatous cells. Collenchymatous cells just below the epidermis. The hypodermis provide the mechanical strength. They provide the mechanical strength to the young stem. In monocot, the sclerenchymatous hypodermis is present. A large number of vascular bundles are also present. The important characteristic of monocot stem, that is sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue is characteristic feature of monocot stem. Here in monocot stem, the vascular bundles are conjoined and closed. Very important. Vascular bundle in monocot stem. So peripheral vascular bundles are smaller than centrally located one. The phloem parenchyma that is absent in monocot stem. That is also very important. Next one, the Dicot Leaf. In case of Dicot Leaf, the characteristics are adaxial. What is adaxial? Adaxial epidermis, that means epidermis in the upper position. Yes, you are right. And abaxial, epidermis, that means lower epidermis. In that case, more stomata is there. In that condition, more stomata is there in abaxial condition. Chloroplast, that is present in mesophyll cell. Mesophyll adaxially placed palisade parenchyma and made up of elongated cells arranged vertically and parallel to each other. And the spongy parenchyma, oval and round, loosely arranged, situated below the palisade cells and extend to the lower epidermis. There are numerous large space and cavities between the cells. The bundle sheath cells in vascular bundles, you know this one, the vascular bundles are surrounded by a layer of thick wall cell. When the vascular bundles are surrounded by the thick wall cell, that is called the, so in between the xylem and phloems are there, and this is surrounded by the cells, that is called bundle sheath, the bundle sheath cell. We found in the C4 plant, can you remember that in Kran's anatomy? Next, the monocotyledonous leaf, in monocot leaf, the stomata are present in both surfaces. That is isobilateral. Isobilateral leaf. Mesophyll is not differentiated. In case of monocot, here the palisade and spongy are not differentiated. In grasses, certain adaxial epidermal cells along with veins modify themselves into large, empty, colorless cells. Can you remember? In case of grass, the adaxial epidermal cell, that is called the bulliform cell. Very important. The bulliform cell. Very important, remember this one. Bulliform cell, what the function? They helps to minimize water loss. Now the secondary growth. The secondary growth. So primary growth is the growth of roots and stem in length with their help of primary growth with the help of their apical meristem, and by the secondary growth increase the girth that are occurred in dicot plants. Tissues involved in secondary growth, that is very important, lateral meristem, So two lateral meristem are responsible for the secondary growth. One is vascular cambium, another is cork cambium. So vascular cambium, it is meristematic layer responsible for cutting of the vascular tissue. The vascular cambium, that is meristematic. Xylem and phloem is there. In dicot plant, the cells of the cambium present between the primary xylem and primary phloem, that is called the intrafascicular cambium. That is intrafascicular cambium. The cells of the medullary cells adjoining intrafascicular cambium become meristematic and form the interfascicular cambium. Is it clear? The intrafascicular cambium, the medullary rays are cells of the medullary rays adjoining the intrafascicular cambium become meristematic and then it forms interfascicular cambium. Now the activity of cambium ring. So can you remember? Intrafascicular cambium or fascicular cambium, the difference you see, intra and inter. When the medullary rays adjoining the intrafascicular cambium, they become meristematic. They become meristematic and form interfascicular cambium. Is it clear, Ankita? Cells of medullary cells adjoining intrafascicular cambium become meristematic and form interfascicular cambium. So in activity of the cambium ring, the cells cut off toward the pith mature into secondary xylem, and the cells cut off toward the periphery mature into secondary phloem. Again I am repeating. The cells cut off towards pith mature into secondary xylem, and the cells cut off towards periphery they are mature into secondary phloem. So everybody, is it clear? So cambium is generally more active in inner side. Very important. Cambium is very active in inner side. Than the outer. So amount of secondary xylem is produced than secondary phloem. Every time the secondary xylem is more than the secondary phloem. During the activity of the cambium ring, primary xylem remain more or less intact in or around the center. At some places, the cambium form a narrow band of secondary phloem in the radial directions. These are the secondary medullary rays. You know the concept of spring wood. The spring wood, in the spring season, the cambium is very active and produces a large number of xylem elements having vessels and wider cavities. Here the cambium activity that is very active, and they form the large number of xylem elements. The wood formed during spring, that is called spring wood, or it is called early wood. Spring wood is also called the early wood, and the autumn wood. In winter, that is in spring, that is in winter, the less activity have formed the xylem elements and have narrow vessels. And this autumn wood is called the late wood. So two kinds of woods are appear in alternate concentric ring constitute the annual ring. So these two kind are from the annual ring. Can you remember? Annual ring. alternate concentric ring. Annual ring that give the estimate of the age of the trees. That helps us to detect the age. Another concept in this arena, that is heartwood. It is the hard, dead, dark brown colored, highly lignified and non-functional central part of secondary xylem. In old trees, the color is dark because the deposition of the organic compound, the dark color. They are dark color. Why they are dark? Because they are having organic compounds. That is tannin, resin, oil, gum, aromatic substance, essential oil. These are present in dark color. And the another is sapwood. The peripheral region of the secondary xylem, they are living and they are light in color. The last component, that is the cork cambium. So cork cambium, meristematic tissue, it is also called phylem. That is phylogen. So phylogen is a couple of layers made of narrow, thin-walled and nearly rectangular cell. There are cork cells are present, that is phylem. The inner cell differentiate into secondary cortex or phyloderm. And the impervious to work due to the sovereign deposition of the cell, that is the cork. So phylogen, phylem and phyloderm. So phylogen, phylem and phyloderm, these three components are collectively called periderm. Remember this one. Phylogen, phylem and phyloderm are collectively called the periderm. There is bark. It's a non-technical term, refer to all the tissues such as the periderm and the secondary phloem. So what are the components in the bark? The periderm and the secondary phloem is there in the non-technical term of bark. Early or soft bark form in the early of the season. Late or hard bark form at the end of the season. So at certain region, phylogen cut closely arranged parenchymatous cell on outer side. These cells are ruptures. You know, these are lenticels. These are lenticels. They permit the gas exchange of between outer atmosphere and internal tissue of stem. So lenticels are developed by breakdown of phelogen. Phelogen breakdown. Cuts off and closely arranged parenchymatous cell in the outer side. They rupture epidermis, form lens-shaped opening, that is lenticell. So we have almost completed all the important topics of anatomy of flowering plants. So I think to get a good revision of this part, now you read the NCERT for the more concise and more detailed. Till then, we meet in the next class with a new chapter. That is structural organization in animals. Till then, goodbye. Have a good day.